Good day, this is Professor Wills, CBMD PhD. It's today Saturday, April 9th, 2022. It is 9:08 and 3 seconds, AM Eastern Standard Time. Today's research, skin cells. So let's hop to it here. Old skin cells reprogrammed to regain youthful function. Findings could lead to targeted approach for treating aging. Date April 8th, 2022. Source, Abraham Institute. The scientists have developed a new technique for rejuvenating skin cells, and this technique has allowed researchers to rewind the cellular biological clock by around 30 years, according to molecular measures, significantly longer than previous reprogramming methods. The partially rejuvenated cells showed signs of behaving more like youthful cells in experiments simulating a skin wound. And this research, although in early stages, could eventually have implications for regenerative medicine, especially if it can be replicated in other cell types. As we age, our cell's ability to function declines, and the genome accumulates marks of aging. Regenerative biology aims to repair or replace cells, including old ones. One of the most important tools in regenerative biology is our ability to create and induce stem cells. The process is a result of several steps, each erasing some of the marks that make cells specialized. In theory, these stem cells have the potential to become any cell type, but scientists aren't yet able to reliably recreate the conditions to redifferentiate stem cells into whole cell types. The new method, based on the Nobel Prize winning technique scientists use to make stem cells, overcomes the problem of entirely erasing cell identity by halting reprogramming part of the way through the process. And this allowed researchers to find the precise balance between reprogramming cells, making them biologically younger, while still being able to regain their specialized cell function. Researchers turned normal cells, which have a specific function, into stem cells, which have the special ability to develop into any cell type. The full process of stem cell reprogramming takes around 50 days, using 4 key molecules. The new method, called maturation phase, transient reprogramming, exposes cells to factors for just 13 days. At this point, age-related changes are removed, and the cells have temporarily lost their identity. The partly reprogrammed cells were given time to grow under normal conditions to observe whether their specific skin cell function returned. The genome analysis showed that cells had regained markers characteristic of skin cells, fibroblasts, and this was confirmed by observing collagen production in the reprogrammed cells. And to show that the cells had been rejuvenated, the researchers looked for changes in the hallmarks of aging. The understanding of aging on a molecular level has progressed over the last decade, giving rise to techniques that allow researchers to measure age-related biological changes in human cells. Researchers were able to apply this to an experiment to determine the extent of reprogramming a new method achieved. Researchers looked at multiple measures of cellular age. The first is the epigenetic clock, where chemical tags present throughout the genome indicate age. The second is the transcriptome, all the gene readouts produced by the cell. By these two measures, the reprogrammed cells match the profile of cells that were 30 years younger compared to reference datasets. The potential applications of this technique are dependent on the cells not only appearing younger, but functioning like young cells too. The fibroblasts produce collagen, a molecule found in bones, skin tendons, and ligaments, helping provide structure to tissues and heal wounds. The rejuvenated fibroblasts produce more collagen proteins compared to control cells that did not undergo the reprogramming process. The fibroblasts also move into areas that need repairing, researchers tested the partially rejuvenated cells by creating an artificial cut in a layer of cells in a dish. They found that the treated fibroblasts moved into the gap faster than older cells. This is a promising sign that one day this research can eventually be used to create cells that are better at healing wounds. In the future, the research may also open up other therapeutic possibilities. The researchers observed that their method also had an effect on other genes linked to age-related diseases and symptoms. The APBA2 gene associated with Alzheimer's disease and the MAF gene with a role in the development of cataracts both show changes toward youthful levels of transcription. The mechanism behind the successful transit reprogramming is not yet fully understood and is the next piece of the puzzle to explore. The researchers speculate that key areas of the genome involved in shaping cell identity might escape the reprogramming process. The results represent a big step forward in their understanding of cell reprogramming, and they have proved that cells can be rejuvenated without losing their function, and that rejuvenation looks to restore some function to old cells. The fact that the researchers also saw a reverse of aging indicators in genes is associated with diseases is particularly promising for the future of this work. Eventually, we may be able to identify genes that rejuvenate without reprogramming, and specifically target those to reduce the effects of aging. And this approach holds promise for valuable discoveries that can open up an amazing therapeutic horizon. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.